So welcome everyone to the Greener Hub webinar series. Once again, my name is Evelyn Zuniga and one of the communications officers for the Greener Hub project. What is the Greener Hub project? The Greener Hub officially started in 2022 as a coordination and support action of the European Commission under the Horizon Europe program. And it currently represents 15 ERANET co-funds and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs last year, and the topic of our first series was circularity in agriculture. Then our second topic was related to biotechnology to advance sustainable solutions for bioeconomy. So if you're interested in topics related to mixed farming systems, mitigation of greenhouse gas emissions, sustainable farming, production of bioplastics, production of green chemicals, among many others, all our recordings are available in YouTube. And now and for these 4 months, we are focusing on a different topic, which is sustainable food systems. And here we're going to have topics related to selling agriculture, food resilience and climate change, food trade, alternative novel food systems, among many others. Just a housekeeping announcement, if you want to type your questions, you can use the chat box or you can use the Q&A box, which it's enabled for everyone. Today, we're going to have two panelists, Dr. Francesco Gai and Dr. Parag Acharya. And give me 1 second because I need to share my screen for our first introduction. Our first presenter is Dr. Francesco Gai, who is a senior researcher at the National Research Council in Italy. He's an expert in animal science, animal nutrition, and metabolism. His research interests are focused towards the quality and food safety of derived products of animal origin. And since 2012, he has been working in collaboration with different research groups and has been involved in the design and execution of multiple research trials on protein insects as potential raw material in fish and poultry nutrition. He's currently the coordinator and partner in multiple transnational European projects. And today, the title of his talk is The Use of Live Insect Larvae to Improve Sustainability and Animal Welfare of Organic Chicken Production, which is part of the Poultry Insect Project. Dr. Guy, thank you very much for joining us today and for accepting to present your project. What I'm going to do now is I'm going to stop sharing my screen so you can start sharing yours and start your presentation. Thank you so much.
Good morning. Thank you, Evelyn, for the presentation and for the invitation to this very interesting webinar. So I'm going to share my screen. Can you see the screen?
Yeah, now we can see the screen. Yeah, now it's in presentation mode. Perfect.
Okay. So good morning, everybody, and thank you to join this webinar. As mentioned by Evelyn, I'm going to introduce our project financed in the SUS Food 2 and Core Organic financing program in the joint call of 2019. This project is recently finished at the end of 2023. So I'm going to give you an overview of our project. But before to go in deep in the project, I want to introduce the topic related to the insect and why the topic of insect is so interesting. As you can see in this slide, the insects are a very, very interesting approach to the circular economy because they can metabolize and they can transform some side streams byproducts that usually are considered as waste, and they can transform from this waste in interesting molecules that have different properties. So you can see there is a protein content, oil, chitin, and frass, and also some bioactive compounds that are chitin, antimicrobial peptides, and also fatty acids. If we go more deep in the what is called the processed animal proteins, that usually is indicated with the acronym PAP, derived from insect, you can see that from substrate waste, there is a mass production of insect biomass that after a step of extraction could be separated. This mass of bioinsect or of insect can be transformed in oil and fats, protein, and novel products. We are more interested in the protein content because there is an application to the animal feed, and there is the fraction that is called processed animal proteins, PAP. Which insect are very vast number of species, but as you can see in the literature in this slide, the most prominent candidates for the utilization of edible insects are substantially the black soldier fly, scientific name is Metzia illicans, the housefly, and the yellow mealworm. And here below you can see the cycle of the reproduction of these insects. And usually the insects for the application in animal feed are collected during the phase of the larvae, because due to the nutritional profile of this stage of the insect. About the legislation in Europe, actually eight insect species are authorized. And in the 2017, the first application was allowed for the utilization in aquafeed of the insect. But in 2021, this was also authorized for other animals, and in particular for poultry and pig feeds. So they enlarged the potential application of insect proteins in the livestock science sector, while the utilization of insect protein is banned in the ruminant nutrition due to the medical problematics. So moving on, in this slide you can see a sort of a summary of which substrates can be utilized for rearing the insect, for the insect production, and which are the target species that can be where the different fraction of the insects can be utilized. So in pet food, aquafeed, poultry, and pig, different fraction of protein, fat, live insects, live insects, that is the topic of our project, and also wool insects. As you can see, the different fractions are not allowed in all the species, so there is some limitation for the moment. So moving to our project, I try to summarize our project in this slide. We have utilized as insect species the black soldier fly that was provided by the Belgian partner, the Inagro colleagues. These larvae were cultivated on agricultural byproducts, and the live larvae were delivered to two different poultry breeds, differing for the grow rates. So we have performed two trials in 2 different years. The first one with the label-necked neck, that is a poultry breed with an intermediate growing period. And the other one is a local breed. It's called Bianca di Saluzzo, that is a typical Italian breed that is considered as a slow-growing breed. So it's particularly suitable for organic farming. And at the end of the trial, so the fattening trial, we have collected different samples, and we have performed different analyses on the physiology of the animals, but also on meat quality of the derived products, in particular the breast of the poultry. And also we have performed some consumer survey and also some LCA, LCC analysis performed by a partner of the project, and the deal there is a German partner. So moving to the experiment design, in the first trial, we have tested the utilization of the supplementation of Hermetia illucens live larvae with a 10% on daily feed intake basis. And we have tested this supplementation both in male and female poultry breed. So in order to verify if there is differences also on the gender basis. And the control was generally a feed, organic feed, produced and suitable for the nutritional requirements of this breed. In the second trial performed, we have tested a control diet that is mainly based on a soybean, and a sustainable diet that where we replaced the soybean with alternative proteins like faba bean and pea protein, so protein that are more sustainable from a different point of view. And in the last one, we have provided the sustainable diet plus live larvae supplementation. But in this case, differing from the first trial, we have provided a 15% of the dry feed intake basis. And in this case is a slow growing breed, so the cycle is more longer, growth cycle is more longer than the first one. So we have fed the animals at least 150 days in order to verify a long-term administration of the larvae. Here there is the composition of the sustainable diet. As I explained, the soybean was replaced in the sustainable diet with field bean meal, pea meal, and barley, and other proteins that are more sustainable. So moving to the material methods and also the results, we have performed, first of all, growth performance and animal welfare determination with different parameters. And I just introduced the results of the first trial that is already published. If someone could be interested, there is the publication on Frontiers in Veterinary Science. And for growth and performance parameters, we have just recorded some statistical differences for the gander, not for the diet. So the supplementation of the live larvae have no impact on the growth performance. And related to the blood parameters, it's interesting that we have differences due to the diet for this enzyme, glutamyl gamma transferase, that is related to the content of a particular molecule of the insect that is called the chitin, that is a part of the exoskeleton of the insect. And probably the chitin have stimulated some parameters related to the lipid metabolism. So moving to the animal welfare, we have performed a different analysis and also some different tests like tonic immobility or avoidance distance test, are all tests that can be provided interesting information about how the animals perceived the supplementation of the insect. And also we have performed, we have recorded some potential damage related to the plumage or to the skin lesion that are indicator of the welfare of the animals. And also we have collected some feces during a different sampling point time during the larva supplementation. And we have determined the corticosterone, that is a biomarker in the skeleton, is a biomarker of the stress of the animals, in order to verify if the treatments with the insect have some effects. As I mentioned, we have also some video recordings in order to check the behavior of the animals. And about some results, it's interesting to notice that from the video recordings, we have recorded that animals treated with larvae have a major exploring behavior. So they exploring the outside part of the poultry facility, and this is interesting because the animals express their natural behavior, that is usually to explore a different in the environment. So this is a positive outcome. Also, another aspect is related to the ground packing. Also, in this case, larvae group in two different sampling points, T3 and T4, showed greater ground packing behavior than the control groups. And also another parameter is the standing. And also in this case, there is a standing over time in L groups. So standing is also another parameter that is correlated with the welfare and to the behavior, the normal behavior of the animals. So here we have summarized what the behavioral analysis can inform us. So an increased exploration behavior in females and benefits, it means benefits in animal management. An increased foraging behavior, it means over time stimulation of a natural behavior repertoire. And also an increased standing behavior, it means that the maintenance of birds activity over aging. So all these parameters are important and are beneficial from the treatment with the insects. Another aspect that we have investigated at the end of the trial, so during the slaughtering of the animals, is related to the gut physiology, and in particular we have investigated the gut histology and the gut microbiota of the animals. Here is summarized the experimental design of the two trials. And here we reported a PCA of the output of the microbiota analysis. So you can see in this slide for the first trial, we have not separate, so the treated and the control group are not very separated, but there is a sort of overlapping. And this probably is due because the larvae have a high water content, so it means that the content of the nutrients are so diluted. What is interesting at when we look at the specific bacterial strain, there is some bacterial strain that are more abundant in treated animals, and particularly is Bacteroides ruminococcus and also Ruminococcaceae. And this is linked, or one of the hypotheses is to related to the content again of the chitin, that is these polysaccharides that could be have a probiotic effect in the bacteria flora of the gut of the animals. So they stimulate to grow some of the strains instead of the others. So in other words, it's a beneficial effect, even if it is a limited fraction of the microbiota. For the second trial, we have differences appeared more differences because, as I mentioned, the control diet and the sustainable diet have different protein spectrum. So here the group are more separated, and there is some differences in the microbiota. Also here you can see the different strains, bacterial strains, and also here you can see that there is some differences in the some strains, like for Lactobacillus and Bacteroides. And this is interesting because there is a sort of protecting effect of the insects, because the Lactobacillus are able to to release some molecules that are beneficial, for example, butyric acid, that are beneficial for the physiology of the gut of the animals. So there is another aspect interesting for the physiology of the animal treated with the insects. So we move to the slaughter and meat quality parameters. So at the end of each trial, we have sampled some parts of the animals, and in particular, we focused our analysis on the breast. That is one of the major muscle of the animal. And as you can see here, there is a slaughtering parameters. A table summarizes the differences. And you can see that for the diet, the only differences for these parameters is related to the spleen that appear to be higher in weight in animal treated with the insects. While for the other parameters like slaughter weight, carcass yield, and so on, no differences appear between control and the treated groups. And also, these parameters are reported in the paper that we published, and I mentioned it before. So another important aspect is the meat quality. So the colleagues involves Norwegian colleagues of Nofima performed some analysis related to the lipid oxidation and the protein digestibility. And as you can see in the slides, there is no differences between control and treated groups, both in raw and heat treated samples of the meat. We have also performed sensorial parameters by sensorial panel, and the test was performed in the laboratory of colleagues of Institute of Bioeconomy of CNR, so here in Italy, and 11 trained judges using a 9-point intensity scale evaluated some different sensory attributes of the cooked breast meat of the animals, and for all the sensory attributes, no differences appear, even if for the color there is an instrumental variation that is related to the gender. So females have meat breast with a higher yellowness. So probably this is due to a physiological effect rather than the diet effect. And finally, we have also performed a consumer survey through cross-sectional data. And this online survey was carried out in the different countries where the partners of the project were involved. So in Italy, Norway, Germany, and Belgium. So we have distributed online survey, and we get around 2,000 respondents. What emerged from the survey is that data from Italy, Norway, Germany, and Belgium relieved significant variations. So because during the question we asked how the consumer perceived a potential meat rising from organic farming or poultry fed with the insects. So the interest was to understand if the consumer are able to discriminate or are favorable in the commercialization and also the consuming of the meat treated with the insects. What is interesting that you can see that the Italian and German consumer show the higher acceptance of poultry fed with insects, while on the contrary, Norwegian and Belgian consumer were more skeptical. Italian consumer exhibited the greatest food involvement and the lowest food xenophobia. That is also a strange or interesting result, because as you know, there is a strong tradition, culinary tradition in Italy. While on the opposite, Belgium showed the lowest tendency to introduce new foods. So could be interesting also to explore, could be interesting for strategy of marketing to introduce in different countries, different strategies to convince or to to promote the utilization of the edible insects in the poultry farm. So I concluded, and I want to inform if someone is more interested to have more information. We have active website, poultryinset.eu, and also we have some social account on Facebook and Twitter. So if someone is interested, of course, they can follow this our project in this website. So I think it's all, and thank you for your attention, and thank you again for the invitation.
Thank you so much, Dr. Guy, for your presentation. It was very interesting. I just want to remind everyone that you can put your questions in the Q&A box and in the chat box, wherever you prefer. So in the meantime, I would like to ask some questions. So are there like any maybe bottlenecks on like the massive production of like the larvae, like like you need to feed so many chickens, and yeah.
Yeah, there is the colleagues from Belgium, from Inagro, have performed some estimation of the production, because of course this is experimental project, so we have not a large scale, but a medium scale or a small scale. But of course, what is important is to have a closest, so not too much distance from the production site of the live larvae or of the insect larvae to the poultry farm, because as you know, what is the ideal is to have the same poultry farm that can be managed also the insect larvae, but of course they know some skill to rear the insect. Of course, we estimated that this type of rearing, in particular for the live insect farming, is suitable for organic farming, that usually are not bigger farms of poultry, so would be a correct ratio of the production of the insect compared to the number of the animals. Of course, it's quite different if we speak about, for example, broiler chickens that usually have, these farms have thousands of animals. So it's challenging to provide enough quantity of larvae for this type of poultry farms.
Okay. Yeah, that makes sense. So yes, makes sense. So we have a question here. It says, how much in kilograms of mass can you estimate to be ideal for feeding 100 chicken layers? Do you have any estimates on the production capacity of BSF output numbers needed to sustain that flock in replacing commercial feed, or it's the same amount as used in kilograms from commercial feed?
So I have to check because if you are interested, we have a deliverable on the website. But of course, if you want to contact me, I can send directly the deliverable where the colleagues have estimated. What I have to underline that in this case, we have not replaced directly the feed of the animals, but is a supplementation. It means that is a sort of environmental enrichment. In other words, if you want to replace totally or partially for example, soybean, you have to utilize insect meal processed animal protein. So it means a protein concentrate from insect. So because of course, as I mentioned, the live larvae are most of the part of the live larvae are water, so the nutrient content is diluted. In other case, if you want to replace for example, soybean meal, you have to utilize a protein concentrate. So you have to dehydrate the larvae and to concentrate the protein fraction. In this case, you have a crude protein content that is comparable with the soybean meal, or even in some cases is higher than the soybean meal content that is around 40%, for example.
Okay. So there's a next question. I also had the same question. Is there like any treatment for the larvae before they are being fed to the chickens? Are they sterilized somehow? Is there like a process?
Usually for live larvae, they are simply separated from the substrates. So are just, I mean, washing, but it's not a washing. Is a sort of cleaning step. But there is not a real sterilization, because the important is to rear the larvae on a substrate that is allowed, and that is, of course, you cannot rear the insects in any substrate that you want. For example, in European Union, is not allowed to use manure to rear the larvae, because there is some health problem, some zoonosis problem. But of course, if you speak about processing protein, of course, there is a sort of dehydration. So there is some step of the extraction. So of course, in this case, there is a sterilization process. But in the case of live larvae, no, just a sort of washing step.
Okay, yeah, great. We have a question here. It says, did you evaluate the heavy metal content of larvae if they are below the EU maximum level?
So this is interesting because, again, the content, possible content of heavy metal in the larvae is related to the possible contamination of the substrates of the rearing substrates. So we have tested the rearing substrates, and we checked that they are below the maximum value. So other problem could be related to the microbiological loads, for example, for Salmonella or for Listeria. And in this case, we have performed some analysis by real-time PCR. And we have found just for Listeria, but not for Listeria monocytogenes. So pathogenic strains. So this is the analysis that usually are carried out.
Okay.
So it's important to check the safety of the substrates before.
Yeah, so on the substrates, they were just vegetable material or also like animal material?
It depends from which type of species. For example, black soldier fly grow well with wet substrates like processed, like ready to eat salads, byproducts, carrots, or other plant-based material that is wet. For example, yellow mealworm grow better with byproducts of cereals, so dry substrates, like, for example, wet bran or other byproducts of the cereal production.
Okay, so it's mixed. It depends on the species. Okay.
Yeah. But of course, you have to respect the legislation, so you cannot utilize, for example, byproducts of slaughtering.
No carcass or anything like that.
Yeah. And this is banned, and I think there is no way to utilize, at least in Europe. In the other part of the world, the legislation is not so strictly like in the European Union.
Okay.
So, for example, I know that in South Africa they can rear substrates on fish byproducts, for example. That is interesting because in this case you can enrich the fraction of the lipid of the insects with omega-3, for example, because they can carry over the omega-3 from the byproducts of the fish. But at the moment is not allowed in European this type of substrates.
Okay, I'm gonna go this last question because we have to move to our next presenter. So it says, thank you for the information. The question is, was the air quality parameter checked? I feel that is a good parameter to be incorporated in production poultry in comparison to WHO standards as AI for global sustainability.
Yeah, I know that for the insect, for example, we checked for the emission of the ammonium, because even if they are mini livestock, they produce some GHG like ammonia. And but for the poultry, if I understood well the question, is the quality parameter checked? Yeah, we checked for humidity, temperature, if this is the parameters that the question is related. Yeah, but is normally checked for the welfare of the animal. It's one of I know the management of the poultry farm. So this is the related to the, but are common parameters. I know are not related to the supplementation of the insect.
Of the food. Okay. Yeah, great. Fantastic. Dr. Guy, thank you so much. It was a fantastic talk. I hope like you keep working in these sustainability projects. They are super interesting, and like for sure more and more people will engage with these kind of practices, and like the consumers will accept more like novel sustainability practices. Thank you so much. Now, thanks so much. Yeah, now we're gonna move to our second panelists. Allow me to introduce, but first I need to share my screen. Yeah, our second panelist is Dr. Parag Acharya, who is Senior Fellow in Food Innovation from the University of Greenwich in the United Kingdom. Dr. Acharya's research interests are focused on developing future feed product and process technology for alternative protein-based food innovation. His research priorities are alternative protein and clean label product technologies by connecting domains of upcycled ingredient technologies, sustainable processing, and novel product formulation. His work aims to understand how novel processing affects the physicochemical properties of alternative plant, algae, or fungal protein sources to deliver the desired functionalities of meat alternatives and dairy-free products. Today, the title of his talk is Climate Smart Food Innovation Using Plant and Seaweed Proteins from Upcycled Sources, which is part of the Ipsos project from the FOSC-ERANET initiative. Dr. Acharya, thank you so much for accepting our invitation to present your project. And I'm going to stop sharing my screen so you can start sharing yours, and the audience is all yours. Thanks so much.
Thank you very much, Evelyn, and good morning, everybody. And thanks, Evelyn, for your introduction. So I will share. You see my screen?
Yes.
Good.
Perfect. Yeah, now it's full screen.
Okay, good. So, actually, this project is funded by CESFood and ERANET. The project is actually ongoing. So it started in 2022 and it will end at 2025 Q3. So the idea of this project, we call it in abbreviative names Ipsos. So innovative upcycle plant proteins for sustainable food systems. Also, this is a multidisciplinary and multi-actor project. So before I move into the project details, I'd like to give a very brief outline, although many of you are already aware about the importance of alternative protein. Now, there are various types of alternative protein currently in the market or also coming up with active research in various countries. One part which is more established is plant and algal protein, whereas the fermentation derived and cultivated meat based on cell culture. These are sort of upcoming technologies which actually a lot of people are looking into, and also there are huge amount of investment that's going on. Now let's try to think that what are the why we would like to go for this alternative protein. As you all know, that if you think of in terms of emissions per 100 gram of proteins, you will clearly see, of course, that the numbers may vary to some extent depending on countries, but overall, in average, the animal protein from animal sources have much higher emission compared to the plant and algal sources. As you can see from this graph, which was compiled based on various studies and also FAO data by University of Oxford. On top of that, another part which is also quite interesting, this is a study from the BCG, the Boston Consulting Groups, and what they found that in terms of emerging technologies, actually plant-based protein has huge amount of not only savings of the greenhouse gas emission, but also if you try to rate it as part of per dollar investment. So in terms of emission or climate smart technologies, this has a huge potential, as you can see from this graph, compared to some of the established sector, like, for example, iron and steel chemicals or shipping. Now, that means this is a kind of untapped climate investment opportunity, and that is one of the reasons that you see a lot of investment currently going on. Now, coming back to these plant proteins or alternative protein as such, these actually enabling the sustainable protein shift, and of course, in terms of the driving aspects, it addresses the climate change, it drives on the sustainable production, and also in terms of reducing food waste, and also provides a growth opportunity. Now, all along the sustainable protein transition, what we also would like to address is basically, I mean, in the current market, if you see in terms of the retail value, there's a huge, around 6 billion market in Europe, and in that aspects, actually, the market is predominantly driven by soya. Yeah. So the question is how we can actually diversify these alternative plant proteins in terms of beyond soya and pea. And I think the whole work of Ipsos is about that, the overarching aim. And then also in terms of the percentage of market share across the continent, you can see that actually Europe is one of the big drivers. Yeah. Now, in terms of the Ipsos project, so what is the strategic relevance of this project? Of course, there are three aspects which we tried to tie in. So one aspect is alternative protein source, and as I said, that how to diversify these plant proteins beyond soya and pea. The second question is coming that can we upcycle the current huge amount of food waste that's happening across the value chain? Can we upcycle that to produce this alternative protein? And in a way that will produce that is in a sustainable manner. So in all these aspects, the thing is that, of course, when we talk about the food waste upcycling, this is all about the circularity, and also it addressed the global challenge that around 2 billion, well, 1.5 billion tons of food waste that is happening, contributing humongously into the climate change, and also in terms of the greenhouse gas emission. Now, whereas in alternative protein, I showed you in the previous slide about the reduction of the greenhouse gas emission, but it also positively influenced the biodiversity and land use change. And in terms of the sustainable innovation, I think this is kind of need of the hour, simply because of the fact that it can actually repair the broken food chain, and also it can accelerate the market opportunity for sustainable products. And if you currently see in terms of the potential of meat and cheese alternatives, which are two of the target product examples that we would like to create through this project, there's already quite a huge potential of like around 4 billion investment that we see in the plant-based meat, and around 1 billion in terms of the plant-based cheese. So in nutshell, actually, the way to more diversify current plant protein portfolio, we can actually use food waste as an important sourcing materials or starting materials. So the Ipsos partners, we have almost around 20 scientists and engineers working together from 7 universities and institutes across 6 countries from 3 continents. We at University of Greenwich leading this project, but at the same time, we also have 2 important industry partners, Beia from Romania and Kanlar from Turkey, and also these scientists and engineers bringing 9 core expertise areas across food technology, food safety, sensor technology, food processing, quality management, and also food loss and waste management expertise. So from that aspects, if we try to think that, okay, I mean, I mentioned about alternative protein, I mentioned about the food waste upcycling. Now, of course, this is not the first project that we are looking into this. There are some previous examples. So we did a sort of scoping on what are the different EU projects, mainly through Horizon 2020 or Horizon Europe. These have been funded. So as you see on the left-hand side, there are a couple of projects already completed or ongoing. And of course, the microalgae or general algal protein is coming a kind of very emerging. And on the contrary, when we started thinking of like planning our projects, we thought, okay, first we need to see what are the focal commodities, in the countries that we are representing. So the 5 countries, because the KBCS from France, they are of course partner, but they are not funded partners. So in terms of funded partners, we try to see what are the focal commodities in these countries. Now, as you see this potato, there is a huge amount of loss happening. And also in terms of the key aspects, which we would like to look, and we actually did a kind of prior arts, a literature search, not much about the processing waste. So potato is in processing used huge amount in starch making. And then in that starch industry, the residual part after preparing the starch, which is actually protein rich, sort of, throw away or used as a landfill. So there is a huge opportunity for that. Now, there are already quite I know one of the UK British company called Brenston from Lincoln, they are already producing this potato protein. But the challenge is the way they produce this protein extract is still a very energy intensive, using a lot of solvents. So the question is, how can we do that more eco-innovatively? Then, of course, similarly, if you think of pumpkin, hazelnut, seaweed, BSG, all these things are either underutilized or underexplored, although containing huge amount of food loss and waste across the value chain. And each of these, of course, the data is a bit, scarce, but still each of these contains quite a reasonable amount of protein percentage. Because in terms of techno-economics, if the starting material doesn't have high amount of protein or the volume is not big, then of course you can think of that we can do a kind of science for the sake of science, but ultimately the aim is to bring it to the industry level to scale up, and that will not be, from the economic point of view, it will not be quite tenable. So we try to zoom in also the food waste, which are quite either they are quite big, large volume, or contain high amount of protein. So the objective, the starting objectives were, of course, to assess the upcycling opportunities for these underutilized protein-rich food loss and waste streams, and then also understand the food safety safety and nutrition aspects of these and develop an extraction, like I mentioned in previously. Of course, there are some plant protein extracts available in the market, but most of them are using quite high energy processing, like wet extraction process, etc. And then the idea was also to actually doing a prototyping of meat and cheese alternatives using this upcycled protein and understanding the consumer insights. So this is the workflow of our Ipsus project. So the starting, assessing the upcycling opportunity of protein-rich food loss and waste, and then bringing to that level of understanding the safety, allergenicity, nutrition aspects, and also the consumer choice. And then that goes into the fact that how we actually extract the protein and using protein, extracted protein as raw materials, understanding the process, function, and quality, interrelationship through some case studies. We would like to do this prototype. And of course, there are in terms of these work packages, there are like certain partners. For example, Greenwich is leading this upcycling assessment work package 1. The École Nationale d'Agriculture in Morocco, they are actually leading the protein extraction part, and Greenwich is leading, say, food safety aspects, and University of Parma leading the prototyping in terms of the innovation case studies and also the consumer stuff. Now, let's zoom into the fact that what the upcycled food loss and waste for the plant protein opportunities, yeah? So, as I said, that, we have these 5 major funded partners with whom we will actually look at these 6 different crops or commodities, yeah, in these countries. And then we would like to see that, okay, how they're. So what we did is that this is actually one of the example you can see from Turkey, but we have actually we did similar studies for UK, Romania, and Italy. The chance is that we couldn't really get a lot of data for Morocco, so for the time being, we left the country for analyzing the data, but the rest of the others we have done analyzed the food loss and waste value chains. And also there are I think more than 15, 16 visits to the various pre and post harvest value chain actors like processors, producers, processors, even the retail. And then try to understand how much how much food loss and waste is happening. Because of course, in either in the literature or some of these websites, you get the data. But I think our understanding of the outcome of this work is that it is very complex, and not only complex, but also many of these data are quite extrapolated, because actual data, the numbers we are trying to crunching, is quite different than some of these reported values, and particularly, of course, when you try to sort of zoom into each of the value chain partners, not as such the overall supply chain. So this is quite interesting, I would say. And then we are also now currently developing some smart toolbox sensor based on the sensor technology for our industry partner Bayer. And that will help us to really, zoom into and very robust and validated way calculate that how much of these streams are being left in the process. So, and then we also looked at the safety and allergenicity aspects of that. And of course, what we did initially is that we tried to do the literature search. And very interestingly, we found that literature, in particular on pumpkin and BSG, the spent grain, is very scarce, very less, compared to that, while, say, for example, the grapes or potato, there are quite a large number of studies happen. And of course, in And then we did a screening of these studies, understanding what sort of information we get. And we came out with 144 publications. And based on that, we prepared a report, which will be culminated into a review articles, literature review articles. And then on the other hand, we also did, one of our partners from Turkey, they did an in silico molecular docking in terms of understanding the allergenicity potential for each of these proteins. And then what we also did actually to support some of these studies with a lab-based work. So we are trying to buy some, which are very expensive, but trying to buy some antibodies and then try to see the binding potential, the actual the wet lab binding potential, and try to match with this in silico modeling. Then comes the eco-innovative extraction of upcycled protein. So our main aim was to first understand the protein content in each of these food waste sources. So in each part of the value chain, like for example, in BSG, we get like 20 to 33%, and it varies across the country. Even within the country, different regions, it varies. And also the type of grains, they use for brewing. I mean, that also varies. So it's not the one type of grain. So there are a lot of variables in this part. And also, like, for example, potato skin, which is another very high volume, left out or food loss, that we try to see that protein content, then seaweeds, etc. So there are two major studies have happened. So at Greenwich, we did a protein extraction, a novel protein extraction from seaweed, which clearly showed that if we actually, introduce a certain pretreatment like ultrasound or high pressure homogenizer, then we can improve the extraction yield quite substantially. And similar things also did by the Moroccan team and the pumpkin protein extraction. And we are just preparing these two results in a form of a paper. So, and then we also looked at, so the University of Parma, they actually started looking at the market and consumers. So it's very important and interesting part, because of course the market trends and consumer perception, that actually, and as well as the intervention and barriers across in different countries, that will actually help us to understand the perception and acceptance of the consumers. And another very important part that we will also be covering in this study is about the willingness to pay. So if we have an upcycled protein, and if we have a claim regarding, the safety and everything, are there consumers who are willing to pay for these ingredients? So we did a state-of-art market analysis, like what you see here, that a number of plant-based meat products in last 3 years. I mean, France, UK, Germany, Netherlands. I mean, these are 4 top countries. A huge amount of new products came into the market in retail chains, particularly, and also with the claims on environmental aspects, on the animal-free aspects, and also allergenicity and free, dairy-free or meat-free, those aspects. We also have done now the focus group studies, which has completed, and then currently we are almost ready with the quantitative or consumer study on that. So we are hoping that will come out with a very interesting results, like particularly I said, like willingness to pay, and also understand the perception of preferential, acceptance of these new proteins. Then I also wanted to give you a bit of brief flavor about the key learning and challenges. As I said, this project is ongoing. Yeah, it has not finished. So there are more results, more information, more outcomes coming. But so far in the journey, we actually learned quite a lot. The first and foremost thing is that we also saw that the stakeholder interest in this upcycling or value addition opportunities is also varies across the countries. For example, in certain countries, the production people, companies, they are not at all interested to diversify to these new ingredients. Also, we saw there are quite a huge amount of difference in the stability aspects of that, which is very important because, of course, we can do a study, lab-based study on understanding the protein, extract the protein. But you have to understand that if we, in terms of the future growth, if we make a manufacturing unit of protein production, and if that is far from where these, food loss and waste happening, then we also need to check that how stable these biomasses are till it comes into the production level, because that relates to the safety and safety aspects of the food. So I think the stabilization is very important. Then also in terms of food safety, like I said, that there are not much studies happened with some of the side stream that we are particularly targeting, and also a little bit less attention to the food safety aspects, both from the ingredient side, so protein sides, and also product side. There are some coming, but not much as it should be. And then also it's huge opportunity in terms of extraction of the plant protein from two aspects. One is the environmental aspect. So can we reduce the environmental footprint of current conventional extraction process? And the second thing is that can you make an economy of scale? Because currently soya protein is sold by 2 to 3 euro per kilogram. Now, question is, can you actually competitively, can you compete with soya protein if we have a particular extraction technology? That we also would like to look at. And then, of course, the claims that I mentioned about, and also the new products that is coming into the market. And there are certain challenges. And I think the two major challenge, I'm not going through all these, but two major challenge in terms of food loss and waste, there is very limited objective data available, and it's very, very difficult to get a very concrete, robust, and validated data because it's so varied, it's so so many different stakeholders are there, or different production units are there, and it varies in terms of quality, in terms of amount, in terms of approachability, etc. And then another very important part, important challenge what we saw is about the lack of quantitative data on upcycled protein, particularly the quantitative consumer data on upcycled protein-based products. And so I think these two are the key challenges that we face. And then before I end, the Ipsos project outcome, I would like to say that I'm heading this Medway Food Innovation Center at University of Greenwich, which actually leading this project, and we are involved not only this project but also different other projects in terms of the Rubisco underutilized legumes, and we also work with we have a food accelerator program where we work very closely with companies, and these state-of-art facilities is helping us in terms of a lot of work that we are currently doing, along with the support and facilities from the partners. So with that, I would like to thank you. And any question?
Okay. Thank you, Dr. Acharya. Thanks so much for your presentation. It's really nice to see how many projects are trying to see how can we take advantage of like the waste products that we all are generating all the time. Maybe we have time for 1 or 2 questions if people from the audience have questions. Then in the meantime, I have a question here. So did you see any challenge associated with the different countries that were involved in the project in terms of like cost implementation and production and like?
I would say yes. I mean, cost implementation, definitely, because of course, in Europe, it's much higher cost, cost of production. But at the same time, I think also to get a good quality of raw materials in many non-European countries is a big challenge, which we have faced. Because, I mean, when we visited some of the brewers in, say, for example, in Turkey, or in other parts, I mean, we have seen that the treatment of the spent grain is very, very different. And I'm saying different because in a way that this is not the way that in UK we had seen, when we visited. But at the same time, I think the main point is that we need to understand how does that impact on the on the stability and safety of these products if we would like to use that for downstream processing.
Okay, we have one question here from the audience. It says, Dr. Acharya, are you planning to do life cycle assessment? And if so, how? Are these novel extraction processes do not exist in the LCA software databases?
Very good question. Thank you very much. Yes, of course, we would not be able to do everything because, it's a huge, we realize day by day that, of course, we propose that in the project, but we realize that each of these work activities are so diverse. So what we intend to do is that we will collect, because we are already collecting data on the wet extraction, which is a conventional extraction, we call it, yeah. And then we also, because as you saw in my slide, that we have introduced a very small changes, like a pre-treatment step, yeah? Now we are also collecting the whole energy, water usage, everything, all these small steps. And then what we would like to do, ideally, a LCA, and then compare that with the yield and the cost. But I don't know, to be honest, like how much we can actually derive the cost, because cost depends on the scalability. This is a very small lab scale things, but at least we can, and I think the energy and other things also varies that way, but at least it can give you a kind of, indication, an indicative results, like, okay, if we have, say, high pressure homogenizer, introduction of high pressure homogenizer to improve the extraction yield of seaweeds, then at what, cost of energy, yeah? And I think that will give you a kind of a trend.
Okay. Okay, perfect. Thank you so much to my two panelists, Dr. Guy, Dr. Acharya. We need to conclude. I'm past 10 minutes. I'm so sorry. Thank you everyone for joining, for your questions. There are more questions. Maybe I can grab them and pass them on to our researchers. Thank you everyone. Thanks so much. And then, so this is it for today for the webinar. Thanks so much. Enjoy the rest of the day. Bye-bye.